So you get a blood test, right? We've all been there. But have you ever stopped to think about what happens to that little vial of blood after it leaves your arm? It actually goes on this whole hidden journey. Before any of that fancy high-tech equipment even gets a look at it, it has to pass a super important inspection by a team of unseen gatekeepers. These folks are the lab's first line of defense, making sure the results that come back are not just accurate, but more importantly, that they belong to the right person, you.
So, you might think every single sample just gets waved through, no questions asked, right? Well, the answer is a big, fat no. It's actually kind of surprising how many things can go wrong between the needle in your arm and the analyzer in the lab. And that's exactly why labs have a really strict set of rules to decide what makes the cut and what gets tossed out.
Okay, so let's talk about that first hurdle. This is all about the absolute basics, the fundamentals that a sample has to meet just to even be considered. Think of it like the very first security check at the airport. If you don't have your ID and ticket, you're not even getting in the door.
I mean, just look at this list. At first glance, they might seem super obvious, right? But each one of these is there to prevent a potential disaster. An unlabeled tube? Come on, you literally can't be sure whose results you're reporting. A leaking container? That's a huge biohazard. It means the sample is junk. And an expired tube? The special chemicals inside might not work correctly, which could totally mess up the results. These aren't just picky little rules. They are the absolute bedrock of patient safety.
And then there's another major red flag, something called hemolysis. This is basically when red blood cells pop. Yeah, like millions of tiny little water balloons bursting open right inside the test tube. And all that stuff inside, it spills out into the surrounding plasma, completely changing its chemical makeup. This kind of contamination messes with dozens of different tests, pretty much making the sample useless. It's an automatic fail, every time.
All right, so if a sample makes it past that first physical inspection, it's not home free yet, not by a long shot. Now it enters the next phase of checks, and this is where things get really precise. It's like a race against the clock, and you have to follow the recipe perfectly.
You gotta understand, these tubes aren't just empty vials. They're preloaded with exact amounts of chemicals, or additives. It's a precise chemical recipe, and the fill level is everything. Take a look here. For a blue top tube, which is used for clotting tests, you've got a little wiggle room, about 10%. But for a black top, used for something called an ESR test, nope, it has to be filled perfectly to the line, too much blood or too little, and the whole recipe is ruined. The test is completely invalid.
And you know what's just as crucial as volume? Time. The clock is always ticking. As you can see on this table, different samples degrade at different speeds. A complete blood count, a CBC, that's good for a day at room temperature. But an ESR test, you've only got 4 hours. But then you get to the prothrombin time, or PT test. It's stable for a day at room temp, but just look at that refrigerated column. Always reject, no exceptions. And that begs the question, doesn't it?
Yeah, why is that? I mean, usually putting something in the fridge preserves it, right? So why would a cold PT sample be an instant no-go? It feels totally backward, but there's some really cool science behind it.
So it all comes down to this one little guy, a protein called clotting factor 7. See, this particular factor, it just doesn't like the cold at all. When you stick that sample in the fridge, the low temperature basically tricks factor 7 into waking up and getting to work way too early. It essentially jumpstarts the whole clotting process inside the tube before the test has even begun.
And that one little thing sets off this whole dangerous domino effect. The machine is designed to measure how long the blood takes to clot, but since the process already got a head start in the fridge, the result is artificially fast. The machine spits out a number that says the blood clots faster than it really does. This could lead a doctor to think a patient's blood is fine when it's not, which could be a really, really bad call. It's incredibly dangerous.
Okay, so we've spent all this time talking about these super strict black and white rules, right? Unlabeled, rejected. Wrong temp, rejected. But you know, in the real world of medicine, for every hard and fast rule, there's almost always a powerful exception.
So what do you do when a sample is just too precious to throw away? What if that sample came from a tiny newborn or was collected during a really tricky surgery? Getting another one isn't just an inconvenience. It could be downright impossible or even harmful. This is where the lab has a special category, precious samples. And this isn't just a casual term. It includes stuff that is super hard or invasive to collect. Spinal fluid, bone marrow, tissue samples, and, critically, any sample from a child under 5 years old. And for these, the rules completely change.
And this right here is where the entire playbook gets thrown out the window. Just look at the difference.
On one side, a regular adult sample comes in unlabeled. Boom, rejected, no questions asked. The doctor gets a call to come and draw a new one. But then, a precious sample from, say, a 2-year-old shows up with the exact same problem. They process it anyway. They run the tests, they release the results, but they slap a big, fat warning label on it, making sure the doctor knows exactly what was wrong with the sample from the get-go.
And this isn't some secret handshake or unwritten rule. No, it's written right there in the official protocol. The main goal flips from enforce the rules no matter what to get whatever useful information we can from this because getting another one is not an option.
Just to give you a sense of scale, right? For a standard adult blood count test, the lab needs at least 1.2 milliliters of blood to do their thing. That's the bare minimum.
Okay, got that number in your head? Now look at this. For a pediatric microtainer, they can work with just 250 microliters. That's not even a quarter of a milliliter. It's an incredible difference, and it just goes to show you the lengths these labs will go to when a sample is just too important to lose.
So what this all really boils down to is the lab's true challenge. It's a constant, high-stakes balancing act. They've got to uphold the toughest scientific standards on one hand, but on the other, they have to work within the messy, unpredictable world of real-life patients.
And this idea, this balancing act between strict rules and messy reality, it pops up everywhere. A fantastic final example? Diagnosing something life-threatening, like malaria. So, for malaria, the perfect scenario, the textbook answer, is to make the microscope slide within 1 hour of the blood draw. But what if it's late? Do they just toss it? No way! The risk of missing malaria is way too high to just say, "Oops, too late." So they make the slide anyway. But they add a special note to the report, letting the doctor know the delay might have changed how the parasites look. They prioritize getting an answer, even if it's not a perfect one.
And that right there is the real mission of the lab. It's so much more than just pushing buttons on a machine. It's about navigating all those tricky gray areas, holding the line on scientific rules when they can, and knowing exactly when and how to bend them when a patient's health is on the line.
Which kind of leaves us with one last question to chew on. We've seen how complex the lab's job is, with all these rules and exceptions. But is their part the hardest? Or is the real challenge out there at the bedside, with the nurse who's trying to get that one perfect, precious sample from a crying baby or a really sick patient in the first place? It's something to think about for sure.